Hello again. My name is Luciano Lucas. I'm the EVP here at DR Vision, and we are here today for the launch of Aivia 5.1. Thank you very much for taking the time to join us today. It will be a brief presentation, probably 35 minutes. Includes 3 demonstrations, and that's why it takes a little bit longer, and a few slides that give a little bit of perspective on why we are doing what we're doing, and also we will cover the main features of Aivia 5.1. So first, we will start with a brief description of why we're doing what we're doing. Then, what are we actually doing? How does it work? And at the end, we will have some information about a promotion that we have for a giveaway. There will be time for questions and discussion at the end. So if you want to stay back and have a little bit more time to discuss things in more detail, you're very, very welcome. I'd like to start by thanking the team that has built this software. This is the full team. Has grown a little bit since 5.0. The full DRV team is even bigger than this, but not everybody's working on Aivia right now. So in particular, I'd like to thank Tuan Fang Chi, who is both of them are the managers for the software and algorithm teams, and Hoyen Lai, who is the product manager and has made a huge effort to make sure the whole thing actually works together. The rest of the team here also contributed very, very significantly, both the software team, the algorithm team, and my own team on the marketing and product management side. Also, I'd like to take this opportunity right at the start to give credit to the authors of the images that we will be using today. From the left to the right, or top to bottom, we have this image here. Comes from Gabriel Martins at the Instituto Gulbenkian of Science in Lisbon. The image in the center comes from Margaret Davis at NIH. And the image on the right, it comes from Professor Martone, who is currently at the UC San Diego. So thank you very much for those images. They really help demonstrating the features that we have developed in 5.1. So the brain is, by most accounts, the organ that is least understood currently, and there is a reason for that. Not only the complexity, but the difficulty in accessing it makes it hard to actually dissect the function, and that's a significant problem nowadays. We realize that we need new tools for studying the brain and understanding diseases related to the brain. And these new tools, some of them will be related to looking at the morphology, the location of the cells that compose the brain. And there's 86 billion neurons of different types and different layers, different locations, but there's 86 billion neurons in a human brain, and about 85 billion other cells that are not neurons. In a small space, so 1 cubic millimeter of data captured with the electron microscope, this converts to around 2,000 terabytes of information. And if we do a whole mouse brain, that's 60 petabytes, or a human brain would be 200 exabytes. That's an enormous amount of data. Not only this becomes problematic moving the data around and handling it, but also extracting meaningful information out of this enormous amount of data is an enormous bottleneck. So there is ongoing effort by multiple groups to optimize the workflows, from sampling the images all the way down to extracting meaningful information. And this is important, not only because we, as scientists, we want to know more about how the brain works, but also because the diseases that affect the brain and the disorders that affect the brain affect a lot of humans. The latest estimation from the World Health Organization is that it affects 1 in 6 humans worldwide. And in that same report, it shows that almost 7 million people die of neurological related diseases each year. And so with relatively little understanding of this organ, we have great difficulty in designing new treatments that help patients that have this kind of disorder. So in recent years, these are the two references, so the Nature 2013 and the World Health Organization report from 2007 cover that information. In recent years, there has been a big push nationally and internationally, and in most of the cases, multidisciplinary approach to study the brain and to map the brain. Each of these projects listed on the top here are very large, multi-million dollar projects, and the ones on the bottom are active, interesting projects that I'll talk a little bit about in a moment. So these on the top all aim to map the brain in some way. The first one is a European Union funded project called the Human Brain Project. And the second one is the Human Connectome Project. This is the Brain Initiative, very well known in the US, but now it's international as well. And the Brain Project, that is a Canadian funded project. And finally, Brain Minds, that's the Japanese initiative for studying and mapping the brain. There's also a Chinese Brain Project that has come online in recent years. And there's also a lot of funding and a lot of researchers involved in this project. So all of these together have a funding of over $1 billion US. In fact, it's closer to $2 billion US now as the projects progress and the funding increases. And this signals an important factor is that these projects are actually trying to develop, in most cases, are trying to develop new tools and new workflows for handling this data. And on the part that touches our software, Aivia, the key bit that touches our software is our tools related to mapping neuron morphology and tracing neurons. And there is a very interesting project led by Hangshuang Peng here in Seattle. He's based at the Allen Institute, called the Big Neuron. You should check it out. It looks very, very interesting, where they test a huge range of automatic neuron algorithms, and from there we can also learn about which algorithms work best and which approaches work best, and then we implement those in Aivia. So that's the Big Neuron. The other project on the far right on the bottom, iWire, is now relatively well known, but if you didn't know it, you should also check it out. This is a game. It's a crowdsourcing game where you manually trace neurons from serial EM datasets. And those projects, both of them, are very interesting because they create ground truth, which later on in those projects, themselves or other people like DR Vision, we could use that ground truth together with the raw data with a different type of technology, not the one I'm talking about today, but we could be using deep learning to train models that could automatically segment images of this type. But that's for another day, another discussion. So there is relatively little understanding about the brain and how it functions. There is also a realization that we need to invest as a global community in these projects that both develop new technologies and then use these technologies to map the brain. And the funding is there from multiple governments around the world. And we, DR Vision, are also trying to contribute to this effort to trace more neurons more automatically and more quickly. And we also have our machine learning tools that help then extract meaningful information out of the traces that we generate. So what are we going to be talking about today, specifically for Aivia 5.1? These are the four major categories of features that we have developed this time around. Firstly, we have significantly improved our neuron tracer. So it includes a few more capability to analyze a few more types of data sets, and I'll discuss that in a moment. We also have a brand new recipe that can detect cells or other objects in 3D space. And this is not necessarily related to neurons, could be anything else, could be just a HeLa cell in a culture, or it could be cancer cells in a spheroid. Really could be anything in 3D, and it could be as big as a whole organism like an embryo, or as small as a small protein foci. Then I will look at our latest update on the machine learning phenotyping, which now includes the potential to use spine morphology also for creating a classifier, as well as branches of dendrites and 3D objects that we create in Aivia. And finally, give you a rundown of some of the major under the hood improvements, mostly to do with speed improvements on multiple parts of Aivia. So I'll cover those today. So first of all, the neuron reconstruction portion. We now cover a wider range of cases. And so the wider range includes detecting automatically the spines. Previously, we could detect, so with Aivia version 5, which we launched about a month ago, 2 months ago, we could detect the dendrites, and now we can detect the dendrites and the spines, which is new. The new version 5.1 that we're launching today can also detect the dendrites without the existence of a soma or somata. That happens when either you have cropped a small region that only has dendrites, or it may even be that the somata are in a different layer that is not of interest to your study. So we can now trace the dendrites without the existence of somata as a starting point for the tracing. And finally, we can also separate somata that are touching each other. So when you have very densely packed population of cells, of neurons, sometimes they touch each other, or they look like they touch each other. And now we have an algorithm that will also separate them. And so you get a more accurate detection. In terms of editing and manual detection, we've also added quite a bit of functionality. You can now detect a soma directly on the image. You can go to the location and click once on the soma, and that will generate the soma locally with a single click. And it's also possible to add spines manually in case one or two or a few didn't get detected by the automatic algorithm. You can then go in and just add them by hand. Or in case you want to delete one because it was wrongly placed, you can delete it and then add it in the correct location. The diameter and centering of the manually drawn dendrites is now done automatically. Previously, we could already draw in 3D using the dendrite pencil, but if you were to draw outside of the dendrite, it wouldn't recenter it, and that caused some frustration. So we've added some functionality to recenter the dendrite trace and also adjust the diameter in real time. Wanted to mention a little bit about the algorithm that we're using, because this is for the tracing of the dendrites. This is probably the most important part of any workflow, is the type of algorithm that you use. This particular one is from 2009, and we find that it is extremely robust at detecting dendrites in 3D space. And one of the really special features of this algorithm is that it can detect dendrites going into that become very, very thin and very dim. It's extremely robust at doing that. So it can trace dendrites into areas where there is very little signal or where the dendrite has become very, very thin. So the technique is called voxel scooping, and a little bit like ice cream scooping. It starts from a starting point, in this case a seed, which can be the soma, or it can be something else if we don't have a soma, and so we cover both cases now. In the first instance, it extracts or it samples a small amount of voxels around the seed point, and it does that incrementally until it starts to find a dendrite or an area that is long. Each time it scoops, or each time it samples a group of voxels, it looks at that group of voxels and calculates the mean intensity for that group of voxels, and if it is above a certain level that we can specify in the software, it assigns it to dendrite, and if it's below a certain level, it assigns it to background. And if it's between these two, it can still assign them to either background or to the dendrite, depending on the offset value that you set. So it's really, really robust. Also at detecting branches and cross points. This is how it looks when you open it in Aivia. It has a simple interface with 3 sections. If I expand the 3 sections, so that's compacted, that's expanded, there are then 3 areas where you can turn things on and off. We have Soma detection is the first section, then Dendrite detection is the second section, and spine detection is the third section. You can turn off Soma detection in case you don't have somata. You can also turn off spine detection if you don't want to detect spines. The dendrite detection is always on by default, and there's no way to turn it off. Then you can see there the key parameters that are needed. I already mentioned them. The intensity threshold, minimum and maximum, the offset, the minimum branch length, and diameter, a dendrite diameter. In this snapshot, it's in pixels. If the image is calibrated, you will see there whatever is the calibration unit that you used, so microns, for example. Okay, so I think now I'll go into Aivia for the first time, so you get a view of how it looks. We just check if everything is going okay. Are there any questions in the meantime? Nothing to answer? No questions? Great, so I can just go here. So I hope you can already see Aivia. I'll just rearrange my window a little bit. Don't need to see me that much. I can leave it there. Yeah. Okay. So if you're familiar with Aivia, this will all look very familiar to you. Perhaps the thing that you will notice is that it's very dark. Until now, we've only had the light theme, which is good if you're working in a room which is very bright. But we now introduce the dark theme, which is very comfortable when you're working in a room that has dim light conditions. So in a microscopy facility, often people will do image analysis in the room adjacent to or even inside where the microscopes are. And in those rooms, typically there is relatively low light. And so to avoid eye strain, we've added this theme for your comfort. Okay, so this is one of the data sets that I mentioned earlier in the credits. The cell, this is a neuron, it's a mouse spiny neuron. It has been labeled with the fluorescent tag, so the dendrites and the spines are labeled, as well as the soma. And we can run here. If this is the first time you see Aivia, you will maybe be surprised that there's a lot of other things we do. We have actually 15 different recipes, but today I'm going to focus on just 2. It's the neuron analysis, which we just looked at in the PowerPoint. So with this data set, I already know the parameters that work, and since this is a demonstration, I don't want to go back and forth too much. We have other webinars that go into more detail and are more like a tutorial. So for today, I won't spend a lot of time explaining how to optimize this, but we do have other webinars. If you're interested in joining, you can join them. We have the Voxel series webinar, and we have the Insight series webinar. So that is a little bit more detailed. So I'm just adding here the parameters that should work. So Soma, get medium, 12, 20, 12. So initially I will turn off the spine detection, and I'm just going to run the analysis for the neuron detection. So I'll start now. I'll hit button now. And this normally is very quick. It shouldn't take more than 20 seconds or so for the algorithm to run the detection algorithm and then the rendering, and that's done now. As you can see, the detection is very accurate, and even in some locations you would be surprised that it detected the dendrite going into what it looks like background, but it's in fact not. If we increase here the intensity of the dimmer voxels just for us to look at, you can see that there is in fact a dendrite going all the way up there. So that would be how to trace automatically a dendrite. Now I will turn on the spine detection. Yeah, here, and just enter the parameters that are needed for this. 3, 10, and 15. So these are parameters. These parameters are very straightforward for a neuroscientist or for the imaging specialist that would normally do this. The parameters are to do with the Soma size. The first one is Soma size. Second one, size in terms of diameter. Second one is the dendrite diameter, which you can also measure in the software with a ruler, an online ruler. The third one is to do with the branching, so how long each of the segments is likely to be. So we can set there the minimum branch length to avoid detecting too small objects, too small dendrites, which are typically background. So we have that possibility. And then for the dendrite detection, we have the intensity threshold, minimum and maximum, plus the offset. And finally, you have the spine information here, which is to do with the spine head diameter, the spine head volume, and the max neck length, which controls the algorithm so that it doesn't start or try to detect spines that are actually background, like these points out here. If we had no control over the maximum spine neck length, it would try to detect those bits of background down there. Okay, so this is now done. The analysis is done, and you can see that it matches relatively well. This is fully automatic, and this is just to show what the process looks like. We can color things a little bit differently so that it's easier to see. Okay, so we have the dendrites in one color and the spines in a different color, and that would be the process for the fully automatic automatic workflow for the detection, for the analysis of this cell. If we wanted now to know a little bit more about what we just captured, we could go into the spine parameters here and know how many spines we have. So we can just scroll down here. It says we have 292 spines. That's what we have. And we could also use our charts, onboard charts, to look at some of the parameters that we detect. So for the spine would be down here. spine set. Can look at average angle, and here we are. So this is these are the groups, the bins for these spines, and you can quickly see that most of them are somewhere between 0 and 2 microns or pixels in length, but there's a relatively large distribution of them, kind of normal distribution here with a very big group down at this side. Can use any of the other parameters as well for this, so volume would be done like this. We try to change the color of the spines just for a moment. Okay, so that would give you already an idea of how to detect things fully automatically. Now, if I wanted to add a spine here, because let's say one of the spines I couldn't detect it. Let's see if I can find one that was not detected. Okay, looks like almost everything was detected. So I will delete one, or delete a few to show how I could add them back. And so this is what I'm doing now, just selecting one. I'm going to delete it. Delete this as well. So I just deleted these two. There they are. Well, they should be there, but I just deleted them. And now if I wanted to add them, this is actually, it turned all the other things back on. So there we go. So this is the analysis that we were just looking at. I have deleted these two spines and I'm going to add them back on. Go to the neuron editor, add spine. I just click on the head and it will trace back to the Soma. Oh, sorry, to the dendrite. But in fact, it is not showing me that. Oh, I know why. Because it's adding it to the first one. Yeah, okay. So I just deleted that. It should add now. There we go. So that is added. Because I had two groups of spines there, it was adding my new manually added spines to the first group instead of the second group. So that would be the process. You just click on the spine head and it will trace it all the way back to the dendrite. Just gonna move around, see if I can spot any other. So this might be a stubby one, very small. Yeah, it seems like it's actually inside of the dendrite, so we won't trace that one. There's another one that was missing, and so on. So you could go around and identify different spines. So perhaps this is another spine right here. You can identify spines that are missing and you can manually add them. And that's one of the newer, one of the new features in Aivia 5.1. Let's say you have really big problems and you can't detect anything automatically. So I'll delete all of the things that I've done automatically. Let's say you have a super problematic data set and it's nearly impossible to do anything automatic. Or you want to do some serious editing somewhere. So the way you do this, so this is, I deleted everything. I just have the original data set now. You use the neuron editor and you start by adding a Soma to this cell. So I can adjust my local table here. I hit once and in real time I get a Soma to appear here. I can adjust how big the detection should be so that I don't get any of the dendrites in. Okay, something like this. Create Soma. So that's now generating a Soma on the spot. And there it is. That's my manually generated Soma. And I can now continue by adding the dendrites, and then later I can add also the spines. So adding the dendrites manually, also with the dendrite editor, so the dendrite pencil, I start here, and you will notice that there is an enhanced trace. If I don't have this on and I trace, both the diameter, I'll do that so you can see, the diameter and the trace will not adjust automatically. So that's the trace I've just done without enhanced trace, and it looks like that, which is not great. So I will delete this one. I'm not interested in this kind of result, and I will add enhanced trace, and I'll trace exactly the same location. And now you can see that it both recentered and readjusted the diameter for me automatically. So I can continue the process. I don't need to be very careful with this because the dendrite, the enhancement is going to figure out what the diameter is for me. So I can trace relatively quickly. There we go. So that's now a relatively long trace. Let me pick one that actually has some spines, so I can show you then how to add spines. Okay, here is one on this side. Okay, here it is. So I'll draw like this. Oh, that one. By the way, there's a Control Z. So if you want to undo anytime, you can just hit Control Z or the undo button and it will go backwards. So I'm tracing this one that has lots of spines so that I can show you how to add how to add spines. I'll also make sure I connect my segments. They're connected, and now I'll add the spines to this segment. So here is one spine. Here is another one, and so on. I think you get the idea. You can then add add spines manually excuse me, to this trace. The reason these are all different colors is because I have not yet joined them, so I can join them very easily. Okay, so these are all connected now. And so that's how you would do some pretty heavy editing if you needed to. For some reason, the automatic tracing wouldn't work. That's what you would do excuse me, to get the best possible results. Okay, so I think I'll go back to the PowerPoint now and move forward. I hope I've covered everything there that was important. Okay, so the next part is related to, oops, just a moment. Okay. So the next part is about 3D cell count or 3D object count in general. The demo will be using nuclei, but it could be using cells or any other organelles or biological features that are of interest. The algorithm is particularly good at detecting things that are close to a sphere, but it can detect also objects that are oblong to a certain extent, and would be happy to test out some more challenging images with you. It can separate objects that are touching like this, and that was a big it's an important factor because very often the images are either the resolution isn't ideal or the actual objects of interest are touching each other. So we've created a brand new automated recipe for detecting 3D objects in 3D fluorescent microscopy images. It can detect a number of things, as I already mentioned, and there are a number of brand new parameters. We already had quite a lot of parameters related to meshes or surfaces, Now we've added a few more related to this particular group of objects. So the recipe console looks like this. When it's fully expanded, it has a portion related to the intensity and size. That's for the initial detection. And following the initial detection, we do some filtering, which is related to the size range. So we can set a size range so that you don't detect objects that are too small or too big. And we also have a tool that separates objects that are in contact with each other. And we call that the separation factor. And if you set it very high, it will more aggressively try to separate objects that are touching. It will look for small dips in intensity along the intensity line of a particular object. And if you set it very low, it will ignore those. It will basically clump everything together. So you have control with this slider to use a low separation factor or a high separation factor. And then you can use it to detect, in this case I use it to detect nuclei, and that's what I'll illustrate now. So I'll go back into Aivia 5.1 and I'll load that data set over here. So initially I'll load a small crop for you to look at and for us to test slightly different parameters. So you can see how it behaves. So this is a small crop from that bigger data set, which I'll load in a moment. And if I set here the parameters, so intensity threshold, I'll set it to 10. And again, I can measure this using the in the plane view. I can use the cursor and it tells me the intensity measurement down at the bottom here. You will see that there is 2, 33, and 44 that relates to the intensity at that location for channel 1, 2, and 3. And I'm particularly interested in channel 1. So in this location, intensity is 2 in the background, and inside the nuclei, it's 89, around 89. But what really matters is on the transition, so on the beginning of that curve, and it's around 10 or so. That's why we have 10 there. Then object mean diameter 50. That's the number in this case, the number in microns. Then the intensity, the object size, we'll set it to 10 to 1000. And finally, the separation factor, initially I'll set it very low. I'll set it to 5 so that you see what is the behavior. Okay, so I'll run it. It's now running the algorithm. It is still a relatively fast process, but it takes a couple of seconds each time you run it. So you can see here the result is pretty good, but there's still some objects that are merged, like these here. So hopefully, if I set this to a higher separation factor and run it again, that kind of object will split. However, like any algorithm, none of them are perfect, and it may be that you'll find a cell or a group of cells that really can't be separated easily. But that would be the process that you could do, is initially set it to a relatively lower separation factor and then later increase it and see if it's necessary. So in this case, I can't really get that particular one to separate, but this separation factor is now causing trouble elsewhere. So you can see here, these cells are now pretty broken up. So I can set it a little bit lower, run it again, and those small objects that were chopped by mistake are now going to be joined back up, and they will be just one cell. So that's the process you'd go through to optimize, a little bit of optimization of this particular recipe. And I find that it's pretty robust when you have cells that are very, very clumped together. So that's already a little bit better, but we can set it even lower so that it doesn't separate those objects. So I'll run it for one last time, and then I'll run it on the bigger data set so you get a view of the whole of that whole sample. Okay, so here it is. That's probably as good as we would get for this particular data set. As with previous recipes, you can look at information in this scatter plot, or in this case I have a histogram, but we have scatter plots too. So we have surface area versus intensity, for example, or volume. So volume versus surface area, and this is an interactive plot. I'll change this. Okay, so you can see if I select a group anywhere on this graph, it will illustrate which one I'm looking at. Also, if I select it directly from the image, it will also highlight it both in this spreadsheet, which sometimes is hard because it's the dots are relatively small, and also in the spreadsheet below. So these three views are connected up, which is very useful sometimes. All right, so I'll load the other bigger data set so you get a sense of how long it takes to apply. Primarily, that's the reason. And so that's rendering. Okay, so that's what it looks like when it's complete. And there are areas here that I would say are very difficult to say where the boundaries actually are. Like this area here is really very difficult to see where the boundaries are. But still, the algorithm is going to be looking for drops in local intensity. And given the parameters that we offer, it tries to break those objects into a reasonable shape. In areas like this, there's going to be some difficulty. In areas where the objects are a little bit more separated, it will operate in a nicer way and provide more accurate results. But the recipe is here. I would encourage you to try it. It is very fast, and you can use this. You can use it as usual. You can look at the results in the charts, the spreadsheet, but you can also use this in our machine learning classifier, which I will go into in a moment. But I just wanted to show you that you can use these features that I just created here. So in this case, there's 1,800 and something features, 8,000 objects, nuclei, and you can teach a classifier using the objects that we have just generated here. And you could select them from this view, or you could select them directly from the 3D view and assign them to different classes. That's how you would teach this classifier. I'll go back to, oh yeah, good. So the last part of the presentation and demonstration is about the machine learning phenotyping, which is an area where DR Vision has a long history and a number of patents. And we have implemented now with 5.0, we implemented the first version that is capable of learning a particular phenotype or particular classes of phenotypes and then finding them in new data sets that have not been shown to the software. With Aivia 5, we could teach based on dendrite segments and whole neurons, meaning the soma and all the dendrite segments. We can now also teach using the spines, cells, nuclei, or small particles, as well as branches, and that's new with 5.1. So it gives you a little bit more flexibility and a lot more power when we look at spine classification. We can also import more file types. We could already import SWC, into the classifier, into Aivia, sorry, so that we could use that content for classification. So this is so that you can bring reconstructions you've created in a different software into Aivia, and so you can reuse analyses that you've done in the past. You can reuse them now in Aivia to do your machine learning part of the workflow. But what is this machine learning phenotyping, and what does it, how does it all work? I've summarized it in this one slide. We do have a full webinar, which is 45 minutes or so, on our YouTube channel and also on our website. And I took this slide from that webinar, but I can summarize it very quickly here in case you have not come across the concept, and this gives you an opportunity to catch up. So there's really a lot of information, and there's typically mixed information and mixed connections between the different points of information. Using all of the parameters, not just one or two that we know should change when we introduce a disturbance to the model. Let's say we treat some cells with a drug that we know should cause an effect on cell size. So typically people look at one or two parameters that they expect will change, and will track those parameters primarily. Now, most cases, there are many other parameters that are not looked at, simply because it's not practical to look at all of them for phenotype classification. But with machine learning, because it's automated, you can actually use all of the measurements that are extracted. So it does require a little bit of work from an expert user, so that you can teach the algorithm which cells or which neurons are of a particular class, or which spines are of a particular class. But you can do that. That's the intermediate step, and then you can apply the new classifier on all images, and this is automatic and very fast. So the first stage is data processing and object detection. That's what I've talked about until now, either with the 3D object detection recipe or with the neuron detection recipe. That's the first stage. It's data processing and object detection. What the software will also do, Aivia will also do, is upon teaching, when you show Aivia that this particular neuron is of a particular class, it will look at and will extract a number of features. And features aren't really just measurements. They aren't diameter, for example. Features can be to do with the size, the intensity, the texture, and relationships between the size, intensity, and texture. So let's say we have 10 measurements. We might have 100 features for those 10 measurements, because they are combinations as well. So the software will learn those features for a particular class, and when you train, you are creating, in our case, a random forest. Random forest, and I do suggest you go and see the webinar with the full explanation, but the random forest is a combination of decision trees, and each decision tree is created by randomly sampling the data points, and then at each decision tree branch it randomly samples the features that it uses to teach that tree. So individually one tree will have a high error, but if you use a random forest, you're creating hundreds or even thousands of decision trees. Each one of them is different from the other, and as a group they are extremely robust at correctly classifying an object. So that's what we do. We create the random forest when you hit the button teach, and then you can apply that random forest, which is the classifier, to new data sets. Okay, with 5.0 we introduced 3 pre-taught classifiers. Just wanted to remind you in case, or show you in case you didn't see it. We pre-taught the data using neuromorpho.org reconstructions with their permission. And we did it for human, mouse, and rat, and these are preloaded with Aivia 5. In 5.1 they're also there, so you can just load your data set and apply a classifier that we have taught here and validated. You can also augment that classifier, can add your own examples. If you're doing mouse work, you can add examples of Purkinje cells or granule cells, and so the classifier becomes more attuned to the types of morphology that you tend to see in your experiments. So the GUI, you've seen it already in the live demo a moment ago, but the GUI looks like this. And the workflow is really straightforward. First, you detect the objects. Secondly, you can teach which object belongs to which class. You can do that directly in the 3D view over here or in the gallery over here. Next, you teach. So you can then apply the classifier, and there is a button on the bottom right-hand corner that allows it to apply the taught classifier on the data set that is shown or loaded in Aivia. And then you can review the results both in the spreadsheet, which I'll show you, it's over here, and in the 3D view. And we have a mode that colors all the objects that were classified per class. So it's very easy to spot whether or not they have been classified correctly. I have already mentioned briefly this, but I wanted to make sure you remember it. We can load reconstructions created in many other softwares. In fact, we believe we are now covering all of the main file types that are created by other software tools. The SWC is probably the most used one. It's a standard one across neurosciences. So you can load reconstructions done in many other softwares into Aivia. You can also load IV files, WRML files, and ASC files into Aivia, and then you can classify them there. In addition, of course, you can just do the reconstructions directly in Aivia and then use them to augment or create a new classifier. And once you have a new classifier, you can apply that classifier to all your images and have them properly classified. Okay, so I'll go back into Aivia. I'll load the other data set, this one here. That's you have seen this data set before. That's the one I used at the beginning. Just reload. Yeah, so I'll reload it because I've done some edits during the previous demo. So I'll reload it. I have it here. And the process for teaching would be to go, so imagine that you have just loaded this data set from another software, you load it into here, the reconstructions had already been done. That's one possibility. The other possibility is that you have done the processing in Aivia. In either case, this would look exactly the same. You would have the image there with the reconstructions, and next step would be to open the classifier tool. And this is the machine learning part of Aivia. And in here, you can load a classifier that is pre-built. So I have already created one for spines. So I'll load a classifier. It's just here. And it shows all of the spines that are showing in this particular image. So there's lots of spines here, and they are all displayed here as well. And I can select them either from the view or from here if I want to teach. But right now I'm just going to apply this classifier that I pre-taught on this image. So the classifier is loaded. It shows me at the bottom, that's the groups that I have taught. There's Philopodia, long and thin, mushroom, thin, and stubby. And the number of examples that I gave it says 9, 9, 12, 11, and 13. And I will hit this button down here, and that will immediately classify all these spines into different classes. So I'll go ahead and do that. And if we see, there are now these classes at the bottom, and these represent subgroups of the whole population. And to illustrate this a little bit more clearly, I'll turn this off for now and just put the spine, color the spine by class. And you can now see each class has its own color, and I can access the statistics for each class down here. So for Philopodia, thin and long, mushrooms, and so on, they're all classified down here. And I can also use the charts to plot them. So that's how you would apply a classifier. I will now show you how to create a classifier from scratch. I won't apply it again because I've already shown you how to do that, but you can see that now we have this type is one type, there's another one, and we can of course export all of these subtypes to an Excel sheet and do further statistical analysis on these. So if I wanted to create a brand new classifier, I would start by hitting the button on the top here and saying I want to create a classifier. I'll call it 5.1 launch, and I select which type of object I want to use for the classifier, and in this case I'll do a spine, so I'll create, and now I have no classes. I just have the single unassigned class with 304 spines, and I will add the first one, so class 1, I'll call it, and I'll add one more class, class 2. So now I have two classes, they're empty, and I want to assign different spines to it. So this is a little bit cheating because they are already all color-coded. So I will turn off the color coding to make them all the same color and go back into the classifier. Okay, so without cheating. I can now go and select directly from the 3D view. So if I wanted to select this particular spine, so I selected this one spine and I want to assign it to class 2, I have to have it selected. So it's selected here, class 2, and I can hit assign, and that adds this spine to this class. I can go on and do that as much as I want. So I continue to assign. I'm just assigning, not really paying too much attention to the shape that they have, but the correct process should be to pick spines that are of a particular class, that you know are of a particular class by looking at them. So I'll try to be a little bit more careful now. I'll pick relatively thin and small ones first, and then I'll pick relatively large ones later. Okay, these are all relatively small and thin. Here's another one. Okay, and now for class 1, I will select ones that have a very obvious different morphology. And I assign them, and they are assigned to that class. Pick a few more. Given that I'm doing this in a live demo very quickly, I don't expect that the accuracy is going to be as good as the classifier that we created offline. To give you an idea, that classifier took around 10 minutes to create, but then you can use that classifier on any other images going forward without having to spend more time on it. So I just created these two classes with a few examples. So there's 7 examples here and 10 examples here. I'm going to teach, and when I hit teach, is going to create this random forest that I already mentioned. It's already done, and it thinks that it will achieve 100% accuracy for both classes. So you can see for the whole classifier, it thinks it's 100% accuracy, and for each class, also 100% accuracy. That's a little bit overconfident, I think, but still, it's an indication. It's not an absolute, but it's an indication of how good it thinks the classification is going to be based on the examples we've given. Now, if I want to apply this classifier, the one I just built on this image, I just hit apply, continue. It has already done that. If I go to the spreadsheet, there's now only 2 classes, and I will color code again the spines based on class, and that's what you can now see. If I zoom in closely, that's what you can see. So classification has worked relatively well. We have some spines in class 1, some spines in class 2, and they are color coded, and as expected, the thin long ones are of one color and the bulky large ones are of a different color, and that's really what we would expect to see. So with that, I will conclude this part of the demonstration and go back into Aivia for the last couple of slides. There is no more live demo from now on, just some summary and an information about the giveaway of the VR headset. So we've made some pretty large, impressive actually, improvements in Aivia that I will not demonstrate today. Well, the dark theme, you've seen it, that's done. But we've also added multiple object selection modes, which I used a little bit, but I didn't really highlight them. We can now select whole cells, dendritic harbor, segments, spines, and individual items. And it gives you really a lot of flexibility when you're trying to interact with a particular type of object. And it lets you get to the statistical parameters or the measured parameters for that particular object directly, which is really, really useful. And we've made some huge improvements in terms of speed, and this you will only notice when you're actually using the software. So I didn't want to spend time here showing you that it's faster, but it is a lot faster than it used to be. And in terms of rendering large numbers of detected objects, we think this is one of the fastest tools out there. We can now do real-time rendering of millions of objects, both in Aivia on the desktop and in VR as well. And if you didn't know, everything that you saw in 3D, we can render directly in VR. Aivia is all set up to render images in VR using the HTC Vive headset, which right now is still one of the best ones, perhaps the best one. And we have fixed a number of bugs, which some of them avoid crashes, but others actually improve performance. There were bugs that made things sometimes slow. So we fixed quite a number of them, and one of them actually made a significant increase in speed for the 2D cell recipes, specifically 2D cell detection and 2D cell tracking. 30% improvement there, which is really a great result. So in summary, we've shown you today 5.1. We can now do full neuronal reconstructions in Aivia, together with quite a lot of editing possibilities. We can also do 3D object detection and count. The machine learning phenotyping part of Aivia has been further expanded to cover even more types of objects, and we talked about spines, 3D objects, just meshes, and dendrite harbors. And we've made some significant improvements in the speed as well, in general, in the speed of rendering objects. That's what I've just covered. If you're interested in pricing, please email directly Queen Tran. Her email is just there. If you want to download the demo version of our software, which is fully featured, there's nothing missing, you can do it through our website. I just realized that my video is on top of the link, so there you go. All right, so drvtechnologies.com/download if you want to try the latest software, and if you want to test send us images so that we can test them and show you what is possible, you're very welcome to use the links provided on this webpage as well. So that's where you do it. So for pricing, Queen, demo, and test images. So the final thing I wanted to mention is we have a headset that we want to give away. We have quite a few of them, not too many though. They'll probably run out pretty quickly. Which looks like this, has a strap, has glass lenses, and basically you can put it on using your phone. You would put your phone in this little slot, close it, put it on, and basically this is a VR headset using your phone. And to make it even better, we'll process your images and prepare them for VR. So you can send us an image, a 3D image, using this website. So I'll show you the website now. Hopefully is visible. So you can go to this website and follow the instructions. There's very simple instructions. So if you have a relatively small image up to 1 GB, you can load it directly here. If your dataset is between 1.1 GB and 10, then please use the Dropbox link that is here. And if it's bigger than 10 GB, we can also work with it. It's no problem, but you will need to use our FTP server. So just tell us that you want to do that using the form at the bottom, and we'll send you the details so that you can send us those datasets. Then just wait a day or two, depending on how busy we are here. We will process the images and prepare them for VR. And after that, you will get your VR headset like this one in a very neat box, which you can use to look at your datasets and do many other things. There's quite a lot of online content nowadays. I'll just show you some of the examples that we have already generated for VR, and you can also put those VR. So these are generated in Aivia. We have created these reconstructions in Aivia, and you can put them on your PowerPoint. You can create links to your website and embed them. And of course, if you have the VR headset, either this one or another one, you can just hit the VR button, and in that moment the screen becomes optimized for VR, and you can then see it in here or in a headset. So if you're using a mobile phone to look at this kind of picture, you would hit that button, put the phone inside the headset and put it on, and you would have your very own VR headset to explore your 3D datasets. So if you send us, when you send us those 3D datasets, we will analyze them for you, prepare them for VR, send you the VR headset, and I hope you enjoy that. We do have limited supplies, so please go ahead and do that relatively quickly so that you're not then later disappointed. And with that, I would like to finish today's webinar. I really appreciate that you joined us and stayed with us until the end. The webinar will be available to review on our YouTube page as well as on our website. We do have a Twitter and a LinkedIn account, so to follow us so that you can see what we are up to, what conferences we go to, what are the latest updates with Aivia. That's the best place. Out of the two, Twitter is the most active, but the most important items we do put on LinkedIn as well. So thank you so much for joining and staying with us. And at this point, I will take any questions that might have been asked in the meantime.
There are no questions.
All right. So thank you so much and have a good rest of the day. Bye-bye.